We'll start again. So I'm waiting for a few more people. They haven't come in yet, but we'll start. So the question is, what are lipids? So lipids are a broad group of naturally occurring molecules, which includes fats, waxes, sterols, fat-soluble vitamins, such as A, D, E, and K, monoglyceride, diglyceride, phospholipids, and others. The main biological functions of lipids include energy storage as structural components of cell membranes and as important signaling molecules. So, when we talk in terms of lipids, people are scared. They think in terms of cholesterol and something going wrong with the heart, etc., etc., etc. Possible. These things do happen when in excess. And please remember that your lipids, your fats, your triglycerides give you more energy than the carbohydrate that we took a look at last Friday. All right? And there are certain, again, dimensions to lipids which you may know or you may not know. So let's take a look at it. So here we go. You will take a look at the complicated structure of lipids. So you can see it's made up of hydrogen, oxygen, nitrogen, double bonds, and I've also shown a few foods that are rich in fat. You've got a burger, you've got ice cream, and you've got a cake. All right. So there are 8 categories of lipids. Do that, that's better. Okay. Lipids may be broadly defined as hydrophobic or amphibolic, small molecules. The amphilytic nature of of some lipids allows them to form structures such as vesicles, liposomes, or membranes in an aqueous environment. It's amphiphilic. Okay. Biological lipids originate entirely or in part from 2 distinct types of biochemical subunits or building blocks, ketoacetyl and isoprene groups. Using this approach, lipids may be divided into 8 categories. This is just for your information, okay? So fatty acids, glycerolipids, glycerophospholipids, sphingolipids, sacrolipids, and polyketides derived from condensation of ketoacetyl subunits, and sterol lipids and prenol lipids derived from condensation of isoprene subunits. So maybe some of these words are new to you. Keep it at the back of your mind. All right, so you can see the structure is quite complicated. You have a ring, and you've got hydrogen, you've got oxygen, you've got nitrite, and so on. So you've got cholesterol, see the structure of cholesterol. Yeah, that is the base. You've got free fatty acids, you can see it. You've got ceramides, and you've got glycoceramides. And I think some of these words you are familiar with. So fats are a subgroup of lipids called triglycerides. And this is what we usually deal with, and this is what we usually assay every day. When they ask you for a lipid profile, we usually do a cholesterol, triglyceride, HDL, the LDL. These are the 4 most important parts, and then there are others. Lipids also encompass molecules such as fatty acids and the derivatives, including tri, di, and monoglycerides and phospholipids, as well as other sterol-containing metabolites such as cholesterol. Although humans and other mammals use various biosynthetic pathways to both break down and synthesize lipids, some essential lipids cannot be made this way and must be obtained from diet. All right? So I'm sure at this stage you have heard a lot of these words. And like I told you, we have mostly dealt with cholesterol. which scares the hell out of us. Triglycerides, again, equally important, but we don't give so much credence to it. And it is the triglycerides that get us into trouble, more so than cholesterol. Someone wants to come in. You alone there? Nazneen Sitna. Okay, Nazneen, get in quickly, please.
Yes, sir, ma'am. Good afternoon, ma'am.
Good afternoon, we've just started. All right?
Yes, sir. Good afternoon, everybody.
There are a few, there are not many, some people are still coming in. Because Rohaina is in,
Good afternoon.
she's in Lonavala for a Scrabble competition. And Gautam Bhai studies at a camp, he'll try to get in. We've got Parul and Parizada here. I think Meena's coming a little late, okay? So we said fats are subgroups of lipids called triglycerides. And it is these triglycerides that really get us into trouble, okay? Especially with diabetics, we saw it last time. Lipids also encompass molecules such as fatty acids and the derivatives, as well as sterol-containing metabolites such as cholesterol. Although humans and other mammals use various biosynthetic pathways to both break down and synthesize lipids, some essential lipids cannot be made this way and must be obtained from diet. And there are foods that are rich in these substances. And then, especially if you have a heart problem, your doctor will tell you avoid this, okay? Yeah. So we asked the first one, cholesterol. So cholesterol is a waxy steroid or fat that is produced in the liver or intestine. It is used to produce hormones and cell membranes and is transported in the blood plasma of all animals. It is an essential structural component of mammalian cell membranes and is required to establish proper membrane permeability and fluidity. So it has its great importance in our cells. In addition, cholesterol is an important component for the manufacture of bile acids, now keep that in mind, steroid hormones and vitamin D. That's the base. Cholesterol is the principal steroid synthesized by most animals. Okay? And you can see the structure is quite complicated. What are you doing? Okay. So you heard of these. So cholesterol is made up of total cholesterol made of HDL, the high density, the low density, and the very low density. Someone's there. Who is it? Meena. Okay. All right. Meena, you've gotten?
Yes,
All right.
sorry ma'am for the delay.
I think we are on slide 7 if I'm not mistaken. All right. And we're taking a look at the cholesterol. The HDL, high density, low density, and the very low density. All right. And then HDL is divided further into LD2, LD3, and so on, as is LDL. And there are some dangerous bits of the LPA, which we will talk of later. And you got the very, very low density, the smaller ones. It's the same slide. All right, so this is the normal values. So total cholesterol, desirable is less than 200 milligrams per deciliter. It's in millimoles, less than 5.1. Borderline high is 200 to 239, and high is over 239. So we have to keep these values in mind. And when it goes above 200, your doctor will tell you please go on something to lower it besides controlling your diet. And I think the most common drug for lowering your cholesterol is statins. And when you take statins, please be very careful. When you start statin, please have your liver function tests monitored, especially your SGPT, because it is known to affect the liver in some cases. Okay, so then your doctor will give you another substitute. So the question is, what is HDL cholesterol? That's your good cholesterol, right? HDL cholesterol is well-behaved good cholesterol. This friendly scavenger cruises in the bloodstream. As it does, it removes. Yes.
You ready?
As it does, it removes harmful bad cholesterol from where it doesn't belong. High density HDL level reduce the risk of heart diseases,
Can you ready?
but low levels increase the risk. All right. So you have Nazneen, mute. Nazneen, please mute. All right. Now the high density is short for high density. HDL is short for high density lipoproteins. Each bit of HDL cholesterol is microscopic blob that consists of a rim of lipoprotein surrounding a cholesterol center. We'll just give it to you for your interest. The HDL cholesterol particle is dense compared to other types of cholesterol particles. So it's called high density. All right, it's called high density lipoprotein. So it acts as a scavenger. All right, so let's go to the next slide. I'm sorry guys, there are some calls coming at the wrong time. So continue to travel through the bloodstream. Cholesterol has to be transported by helper molecules called lipoproteins. What are you doing?
No, I'm just like
Go back.
yeah, this one. It's doing this one.
What was the other one that you put in? See, do it
Yeah,
and
it is.
Acha, go forward. No, do that. The other one. HDL cholesterol scavenges and removes LDLs. Huh? It's a scavenger. So it just goes out and digs the low density, which is your bad cholesterol, because this cholesterol sticks, and this cholesterol is what gives you plaques. And plaques, as you know, eventually give you a heart attack. Keep it in very simple words. So HDL reduces, reuses and recycles LDL cholesterol by transporting it to the liver where it can be reprocessed. HDL cholesterol acts as a maintenance screw for inner walls of blood vessels, endothelial. Damage to endothelium is the first step in the process of arteriosclerosis, which causes heart attacks and damages and causes strokes. HDL chemically scrubs the endothelial clean and keeps it healthy, right? So I told you LDL is the bad one, it sticks. HDL is a good guy, it acts as a scavenger and gets rid of the LDL. Because if the LDL is allowed to prosper, it's going to give you plaques, it's going to give you clots, it's going to give you heart attack, all right? So arteriosclerosis is a development of this fat, all right? So HDL cholesterol levels greater than 60 milligrams per deciliter are high. That's good. HDL cholesterol levels of 40 milligrams, they're low and it's bad. And this is what your doctor monitors. Please take a look at the pictures that we have below. So you've got cholesterol is a waxy fat carried through the bloodstream by the lipoproteins. And see how the deposition takes place. You've got high density lipoprotein, the good cholesterol. You got LDL, the bad cholesterol. And you can see that LDL is the one that sticks. So we'll take a look at the picture. Yeah, so LDL is the bad cholesterol. Oh my God.
Yeah.
And you take the pump out. Guys, just a minute, somebody's at the door. It's my dog's medicines which have come. Okay, so LDL cholesterol is important for the transport for the process of narrowing arteries called arteriosclerosis, right? So the deposition takes place. Some LDL cholesterol circulating through the bloodstream tends to deposit in the walls of the arteries. This process starts as early as childhood or adolescence.
Can I pay?
No, it's already paid. Nothing. Tell him to go. Guys, just hold on. Just hold on, please. This process starts as early as childhood or adolescence. White blood cells swallow and try to digest the LDL, possibly in the attempt to protect the blood vessels. All this is paid for. Who told you to pay?
I don't know.
He told you to pay?
हाँ बोल रहा हूँ.
क्या?
बोल दिया कि पेड़ है.
Then?
चलो ठीक है. Change कर दूँ.
All right, no. So I will go over this. So we said the LDL is the bad cholesterol. Keep it in mind. The process of being the bad cholesterol. What does it do? Because it tends to stick and forms deposits in the walls of arteries. The process starts as early as childhood or adolescence. White blood cells swallow and try to digest the LDL, possibly in the attempt to protect the blood vessels. That is a function of the white cell, right? Acting as a phagocytosis. And take a look at the picture below. You can see that your LDL is sticking. Look at his face, and you've got the HDL coming to try to wipe him out. It's a very sweet little picture. Can you see? Next slide, next slide में जाओ. ये 15 है.
Yeah, ma'am.
All right. So we've got total cholesterol made up of LDL plus HDL plus triglycerides, and there's a formula to calculate each, right? So LDL is equal to low density lipoprotein, and HDL is equal to high density lipoprotein, and we'll see the structure. You just take a look at it and remember what it's like. So HDL, most important, removes cholesterol from the blood. All right. 16.
Yeah, ma'am.
Slide 16.
Yeah, ma'am. 16 ही कर रहा हूँ. थोड़ा ये काम नहीं कर रहा है.
कैसे काम नहीं कर रहा है?
Oh wait.
You've gone back to the other one.
16, no, 16, 15 का 16 है.
Okay, the same picture. In process of the white blood cells convert the LDL to a toxic oxidized form. More white blood cells and other cells migrate to the area, creating a steady low-grade inflammation in the artery walls. Over time, more LDL cholesterol and cells collect in the area. The ongoing process creates a bump in the artery called the plaque. The plaque is made of cholesterol, cells, and debris. The process tends to continue growing the plaque and slowly blocking the artery. An even greater danger than the slow blockage is a sudden rupture on the surface of the plaque. A blood clot can form and on the ruptured area, causing a heart attack. So this is the sequence of how heart attacks come through. Your LDL is being deposited regularly in the arteries. There's a bump in the arteries. And you've also got the white cells that keep attacking it to, what's the function of the white cell? Phagocytosis. So it tries to remove this little thing called the plaque. Doesn't succeed. And this process continues for quite some time. And eventually, what happens, the danger is a blockage. The blockage ruptures and the clot that is formed rupture in the area, and this is what causes the pain and the heaviness of the heart attack. So this is a sequence of events. I hope it is very clear as to how a heart attack occurs. Very clear? Keep it in mind. All right. So reference ranges are here. The LDL of less than 100 milligrams per deciliter is optimal, and LDL of 100 to 125 or less is near optimal, and LDL of between 130 and 159 is borderline high, LDL of 160 and above is high, and LDL of 190 milligrams is high and it is undesirable. The total cholesterol, the formula is, I think it's not here, HDL less than 4 as a ratio. The ratio of cholesterol to HDL should be less than 4. All right. So we come to triglyceride, and here you see I have this obese man on the right hand side. So triglyceride is an ester derived from glycerol and three fatty acids. Sorry. There are many triglycerides depending on the oil source. Some are high, unsaturated, some less so. They are the main constituents of vegetable oil, typically more unsaturated, and animal fats, typically more saturated. In humans, triglycerides are a mechanism for storing unused calories, and the high concentration in blood correlates with the consumption of starchy and fatty foods. Alright? So, your triglycerides, we try to keep the triglycerides as low as possible. Alright? So, the function of triglycerides as main components of very low density lipoprotein VLDL and chylomicrons plays an important role in the metabolism of energy sources and transporters of dietary fat. They contain more than twice as much energy, 9 kilocalories or 38 kg per g, as carbohydrates and proteins. And it is this, your triglycerides, is the main source of energy, gives more energy than glucose. And you have a very nice little cartoon here. It says triglycerides. Let's just say when your cholesterol, HDL and LDL are parting it up, he is the guy you don't want to get crash. The doctor is telling the patient, and you can see the patient is in the ward, rather in the ICU. Okay? So these are your reference ranges. Post 8 to 12 hours of fasting, the American Heart Association recommends. So you have ideally less than 150. Then you have a danger signal. Normal range, low risk. Okay. Then you have a between 175 and 199, slightly above normal. We don't like it. Then you have a triglyceride of 200 to 499, high risk. And you have over 500, you have a very, very high risk. Now guys, a very good way for you to take a look at the serum and to know that triglycerides are high. In a fasting serum, yours and mine is clear. Now the serum starts getting cloudy. And it gets cloudy because of the triglycerides going up. And you can get triglycerides where the serum is milky. And that is your danger signal. That is your danger signal. Alright? So we as techs, when we have samples from patients for lipids, if we find that the serum is clear, then we say, acha, looks like the triglycerides going to be fine. But if it starts becoming turbid, cloudy, then we worry. Alright? And that's very important. Another thing, please remember why we tell the patient to come 12 hours fasting is because your chylomicrons, very, very low density lipoproteins, takes about 9 to 10 hours to clear up from your blood once you had a meal. Okay, that is the reason why we tell the patients, please come after a 12-hour fast so that your serum hopefully is clear. So even after a 12-hour fast, your serum is not clear, then there is a pathology somewhere, needs to be investigated. So keep this in mind. For lipid studies, we tell them 10 to 12 hours fasting because we want the serum to be clear. We want the chylomicrons, the very, very low density lipoproteins to clear up from the serum. That is how long it takes for it to clear up after a meal. And you've seen after, let us say that we do a 2-hour sample, you will find that the serum may be slightly clouded or the plasma may be slightly clouded because the chylomicrons have not cleared up. It takes about 10 to 12 hours for them to clear up. So keep that point in mind, alright? Yeah. This is just a little extra for you, lipoproteins. We do it on cardiac patients. So lipoprotein is a biochemical assembly that contains both protein and lipids. The name tells you. Water bound to proteins. Many enzymes, transporters, structural proteins, antigens, additives, and toxins are lipoproteins. Example includes the high density lipoprotein, the good one, and the low density lipoprotein, which is the bad one, which enables fats to be carried in the bloodstream. The transmembrane protein of the mitochondria and the chloroplast and bacterial lipoproteins. So you can take a look at the structure. You can see that the HDL is a nice, clear, clean structure. It's got a nucleus and you've got the triglycerides and the phospholipids surrounding it. But can see the LDL? Just take a look at the picture. Okay? Alright, so of course this is a graphic demonstration and we do monitor lipoprotein.
Can I change?
Yes. So we said lipoproteins may be classified, listed from larger or less dense, smaller and denser. Lipoproteins are larger and less dense if they consist of more fat than of protein. They are classified on the basis of electrophoresis and ultracentrifugation. So this is your LDL, lipoprotein. Okay, and you can see that it is quite dense. All right, it's not as clear as the other one. And I'm just illustrating this for you again. A picture of the LDL lipoprotein and the HDL lipoprotein, and you can see that the HDL lipoprotein is a nice, clear sort of a structure. You got triglycerides in the periphery, and you got phospholipids also, and you got the protein in between. Where are you?
No, 23 to 24 slide.
You sure?
Yeah, ma'am.
So we got high triglycerides and diabetes. I think we spoke of that. Triglycerides account for 99% of fat stored in our bodies. These triglyceride-laden fats serve as the most important source of long-term energy for the body, since they are stored in much denser form than starches and muscle proteins. Keep this in mind. The formation of fat requires the presence of insulin. You remember your carbohydrates? Triglyceride in fat is converted to energy between meals and overnight and any time that we are fasting or insulin levels are low. Fat cells have tremendous storage capacity, which may contribute to obesity. With extended fasting or absolute insulin deficiency, the liver converts fat breakdown to products known as ketones, and these ketones are dangerous. Just take a look at this, right? So for the fats to convert further, you need insulin, right? And insulin in diabetics is on the lower side, and we will see what happens. It's conversion of liver. So the body has to pull. The body has to pull for energy. It will pull from carbohydrates, it will pull from lipids, and it will pull from proteins. So monitor exercise and diet lowers the triglyceride and diabetes. And you have the figures in front of you. All right? All right? So triglyceride diabetes process. We'll continue, huh? High triglyceride levels in blood tend to coexist with low levels of HDL. Has to be right. Contributing to a condition called diabetic dyslipidemia. The third component of this dangerous trio is a tendency for patients with this condition to have small, dense, undesirable, more atherogenic type of LDL cholesterol in the blood, even though the LDL cholesterol level may be normal. The combination of high triglycerides, low HDL, and central obesity are the hallmarks of metabolic syndrome, which occurs in 80% of people with type 2 diabetes. The frightening significance of this combination of risk factors is a marked incidence in people of premature death from heart disease. So this is what happens, and you can see the gentleman sitting down there is quite obese. Yeah, so his fat has accumulated and he's putting on a lot of weight. All right.
Okay.
So you have the lipoprotein structure. Chylomicrons. You got ApoA, ApoB, ApoC, ApoE, and T and C, and so on. So you can see where it's green, and you can see the structure. You got ApoA, ApoB, and you have all the other apos. All right. This is just to give you a picture. Lipoprotein structure. Chylomicrons are very, very low density lipoproteins. So what is ApoA? So besides doing triglycerides and fats and SCPK, CPK and B, doctors ask for ApoA. So ApoA1 is a protein that in humans encoded by the ApoA1 gene. It has a specific role in lipid metabolism. ApoA1 is a major protein component of high density lipoprotein in plasma. Chylomicrons secreted from the intestine enterocyte also contain ApoA1, and it is quickly transferred to HDL in the bloodstream. This is just for your revision, something extra for you, all right?
Okay.
So here the biomarker is here. You got IgG, you got transferrin, you got fibrinogen, you got Ig, and you got all the different micromolecules that are needed in the body. Okay, then you got as a major component of high density lipoprotein complex, good cholesterol. ApoA1 helps to clear cholesterol from the arteries. So the sentence is important. ApoA1 helps to clear cholesterol from the arteries. And you can see the structure. You got HDL with ApoA1, you got HDL with ApoA1 delta, and HDL with ApoA1 delta, and you can see the figures. So ApoA1 helps to clear cholesterol from arteries. All right.
Okay.
All right, these are mutants. There's something, is an anomaly, just keep it in mind. One of the 4 mutants of ApoA1 is present in thoroughly 0.3% of the Japanese population, but is found 6% of those with low HDL cholesterol levels. HDL is low, but ApoA1 is present. ApoA1 Milano is a naturally occurring mutant of ApoA1 found in a family descended from a single couple of the 18th century, described in 1980. It was first known molecular abnormality of apoproteins. Paradoxically, carriers of this mutation have very low HDL cholesterol levels, but no increase of risk to heart disease, right? So HDL is low, ApoA1 Milano is found, and there's absolutely no risk to heart disease. And this is found mostly in the Japanese population. And you know how healthy the Japanese are. Their lifestyle, their diet, unbelievably wonderful. And they live to very, very old ages. I'm sure you've read a lot about it. And just keep it in mind. Mutants of ApoA.
Yes.
This is what I've explained to you. Biochemically, ApoA1 contains an extra cysteine bridge, causing it to exist as a homodimer or a heterodimer with ApoA2. However, the enhanced cardioprotective activity of this mutant, which likely depends on cholesterol efflux, cannot easily be replicated by any other cysteine mutants. Combinations of ApoA1 melanodimers formulated with liposomes can reduce arteriomas in an animal model by up to 30%. ApoA1 Milano has also been shown in clinical trials to have statistically significant effect in reversing plaque buildup on arterial walls. There we go. So ApoA1 Milano has been shown in small clinical trials to have statistically significant effect in reducing plaque buildup on arterial walls. In human trials, the reversal of plaque buildup was measured over the course of 5 weeks. So this is wonderful, isn't it? Same thing, and so and Athoma, a fatty deposit within the inside linings of arteries. Lifestyle factors that reduce the risk of forming atheromas. All right. You can see the structure of what happens. ApoA1 Milano infusion induces regression and reverse remodeling of human coronary plaques. So evidently an infusion is given, and you can see that the surface, the inner wall, the inner surface opens up, and the blood flows freely. Okay.
Okay.
So types of lipoproteins. Chylomicrons carry triglycerides, fat from the intestine to the liver.
No.
Skeletal muscle and to adipose tissues. Very fair. Chylomicrons carry triglycerides from the intestine to the liver, skeletal muscles, and adipose tissue. Very low density lipoproteins carry triglycerides from the liver to adipose tissue. Intermediate density lipoproteins, IDL, are intermediate between VLDL and LDL. They are not usually detectable in the blood. Just for your information. Low density lipoproteins carry cholesterol from the liver to the cells of the body. LDLs are sometimes referred to as bad cholesterol lipoproteins. High density lipoprotein collects cholesterol from the body tissues and brings it back to the liver. HDLs are sometimes referred to as the good cholesterol lipoproteins. Alright.
Okay.
So this is the structure chylomicron, and it's made up of the VLDL, LDL, and HDL. Okay.
Okay.
So then we come to ApoA. The ApoA was a good one. The ApoB is the bad one. Apolipoprotein B is primary apolipoprotein of low density lipoprotein, right? LDL, there you go, which is responsible for carrying cholesterol to tissues. While it is unclear exactly what functional role ApoB plays in LDL, it is primary apolipoprotein component is absolutely required for this formation. What is clear is that ApoB of the LDL particle acts as a ligand for the LDL receptors in various cells throughout the body, unlocks the doors of cells and thereby delivers cholesterol to them. So you can see ApoB, the cholesterol is right in the center. It's made up of phospholipids. Alright. This is something extra for you to know. So mechanism of action, through a mechanism that is not fully understood, ApoB can lead to plaques that cause vascular disease, arteriosclerosis, leading to heart disease. There's considerable evidence that levels of ApoB are a better indicator of heart disease risk than total cholesterol or LDL. Keep that in mind. However, primary for historic reasons, cholesterol and more specific LDL cholesterol remains the primary lipid risk or the risk factor of arteriosclerosis. So you got HDL, you can see HDL ApoA1 below, and you got LDL ApoB. So reference range,
Yeah.
where you got?
Just here complete one और इसके बाद ये.
Alright, so genetic variant of ApoB. High levels of ApoB are related to heart disease. Hypobeta-lipopeninemia is a genetic disorder that can be caused by a mutation of ApoB gene and ApoB. Beta-lipoproteinemia A is usually caused by the mutation of MTP gene. MTP mutations in gene ApoB100 can cause familial hypercholesteremia, a hereditary form of metabolic disorders. Hypercholesterolemias. Just an excess information for you. The range of ApoA-B immunotherapeutic method. Reported range of ApoA1, 115 to 206 mg for females, and 107 to 187 for males. It is recommended that each laboratory establish its own normal range. Ranges of ApoB, immunoturbidimetry methods, expected values. Reference range for ApoB are 60 to 150 milligrams per deciliter for females and 63 to 152 for males. It is recommended that each laboratory establish its own normal range. And you have to compare it with LDL, that is what they tell you. Yeah. Yes. Now, this is LPA, and taking a look at the structure, it tells you that something's not right? So LPA is a member of the family of molecules that carry LDL cholesterol, the bad kind, that clogs arteries in the blood. The most common such molecule is apolipoprotein B, which consists of LDL with one protein attached. LPA is different because it has additional protein attached, and it's called lipophosphatidyllic acid, and the structure itself tells you that it's bad, it's fatty, and it's something that sticks. Change. Why are you asking me this word? The pathology: lipoprotein structure is similar to plasminogen and TPA, tissue plasminogen activator, and competes with plasminogen for its binding site, leading to reduced fibrinolysis. Keep this in mind. Also, because LPA stimulates secretion of PA1, it leads to thrombogenesis. In addition, because LDL cholesterol content, LPA contributes to arteriosclerosis, plasminogen A activator, or PPA activator. Okay, so this is what it is. PPA1 is a plasminogen A activator. So now something here happens is it reduces the fibrinolysis. Let's go further. Risk of LPA levels in blood. High LPA predict risk of early arteriosclerosis, similar to HDL-LDL, but in advanced arteriosclerosis, LPA is an independent risk factor for dependent on LDL. LPA then indicates a coagulant risk of plaques thrombosis. ApoA contains domains that are very similar to plasminogen, PGL. LPA accumulates in vessel walls and inhibits binding to PLG, the surface, reducing plasmin generation, which increases clotting. The inhibition of PGA by LPA also promotes proliferation of smooth muscle cells. This unique features of LPA suggests LPA causes generation of clots and arteriosclerosis. So it's important for you to know the last line. LPA causes generation of clots and arteriosclerosis. Okay. So here we go. Normal artery, mild arteriosclerosis, you can see the deposition, severe arteriosclerosis. It nearly blocks your whole vessel wall, isn't it? Who is at risk? Vegetarians have higher levels of LPA than fish eaters. In one homogeneous tribal population of Tanzania, raising the possibility of pharmacological amount of fish oil supplements may be helpful to lower the levels of LPA. It's a nice thing to know. Some studies have shown that regular consumption of moderate amounts of alcohol leads to significant decline in plasma levels of LPA, while other studies have not. But that does not mean that person must go on high alcohol levels. LPA risk table: desirable less than 14 mg, borderline between 14 and 30 mg, high risk 31 to 50, very high risk above 50. LPA appears with different isoforms per Kringle repeats of apolipoprotein. 40% of variation in LPA levels when measured in milligrams per deciliter can be attributed to different isoforms. Lighter LPA are also associated with disease. This thus a test for mg, but the deciliter is not preferred. Okay. I don't want to get into Kringle repeats, but you can take plasma lipoprotein A concentration is related to cardiovascular risk. Keep that in mind. The defined protein component of LPA particles, apolipoprotein A, is encoded in the LPA gene. So just keep this in mind. I don't want to go into this. Two principles methods of detecting variations of KIV1 and 2 repeat numbers are electrophoresis with immunoblotting to detect ApoA protein isoforms, or pulse field electrophoresis of unamplified genome DNA to detect the variation of LPA gene. Just keep this in mind. You can look up Kringle repeat. Okay, this is a picture to demonstrate the Kringle. So, it's homologia, structural, and okay, this is in French. Okay, all right. So, types of lipoproteins and the functions. There are four major classes of lipoproteins. Chylomicrons, which circulate in the blood and deposit the triglycerides and fatty tissues. You've got very low density lipoprotein, which, like chylomicrons, also circulate in the blood and deposit triglycerides and fatty tissues, but contain a higher concentration of cholesterol. Low density lipoproteins, also called bad cholesterol, form 60 to 70% of person's total cholesterol. And high density lipoproteins, also called the good cholesterol, removes excess cholesterol from tissues in the body. Okay, the types of lipoproteins, four major types. Here we go, structure. You can study it. Okay.
Okay.
So we come back to lipoprotein. Lipoproteins are molecules which carry cholesterol and triglycerides throughout the body. Now that's something you can know. They are both vital to body some functions like building cell membranes, hormones, and providing energy to the body. However, an excess of either, in particular cholesterol, can put a person at serious risk of cardiovascular disease. To help measure the type or amount of cholesterol in the body and determine potential risk for cardiovascular disease, lipoprotein electrophoresis is frequently performed. During this type of electrophoresis, the quantity of each lipoprotein class is measured and based on its movement in an electrical field. Okay. All right, now this is something which is not very old. I got this from a professor at KDH. All right, this lipid tetrad, because it's related to our population. This lipid tetrad consists of elevated LPA in combination with lipid triad. This lipid triad is rarely seen in other populations in epidemiological studies. However, the combination is frequently seen in patients with severe cardiac disease and in those that have poor outcomes from repeated PTCA, blood gas, CABG, or heart transplants. Arteriosclerosis in young Asian and high morbidity and mortality from CAD can be attributed to this tetrad. So youngsters have it. A comprehensive lipid triad index has been described as a single best predictor of CAD in diverse populations, especially Asian Indian. This index reflects the total burden of dyslipidemia and is derived from multiplying the three lipids directly associated with CAD and dividing the product by HDL, which is inversely associated this way. So look at the formula. Just take a look at the formula. Cholesterol into triglyceride into LPA upon HDL. Okay. Then you got the lipid pentad index. a novel bioindex for evaluation of lipid risk, particularly for arteriosclerosis in the young adolescents and children of premature coronary artery disease patients in India. LPA, ApoB, cholesterol, LDL cholesterol, triglyceride upon ApoB1. A significant increase in the mean levels of LPA, ApoB, total cholesterol, low density lipoprotein cholesterol, and triglycerides in cases when controlled. In contrast, ApoA1 and high density lipoprotein cholesterol values decreased. Lipid tetrad index, LTI, and arteriogenic index in Indian children and adolescents was also calculated. The Kolmogorov, these statistics and cumulative possibility plot suggests that new lipid pentad index defined is able to discriminate case and control populations more precisely than the existing LTI and arteriogenic index. So this is the formula on top. LPA into ApoB into cholesterol into LDL, triglyceride, and upon divided by ApoA1. The various indices have been used for diagnosis and prognosis of cardiovascular disease. Arteriogenic index of plasma, AIP, is logarithmically transformed ratio of molecular concentrations of triglycerides to HDL cholesterol. A strong correlation of AIP with lipoprotein particle size may explain its high predictive value. Summarized data of AIP calculated in 8394 subjects from 6 populations and clinical studies. So AIP less than 0.11 is low risk, AIP between 1 and 0.21 intermediate risk, and AIP more than 0.21 is increased risk. Now this is something which is new. Just keep it in mind that these two parameters are used to calculate risk in people, especially in children, especially in Indian children. I think that's the last bit. So this is the end. In fact, some bits may be a little complicated, but it is just a little bit that I've thrown at you so that you know that lipids, there is just not cholesterol and triglycerides. There are many, many things that we study with lipids. Anybody? Any comments? You'll have to read this. I tried to make it as simple as possible. I cannot go into each slide. I can if you were in class. But this is just to tell you what it is. Cholesterol, triglyceride, fine. LDL, HDL, fine. But what is the use? What do they do? Where does the danger lie? Why is it that we look into triglycerides very, very minutely? And how do we take a look? We take a look at the serum, and if the serum is cloudy or milky, then we know the triglycerides are high. And one has to be careful. Anybody else? Any questions? Everybody's too quiet.
It was nice, man. It was very in-depth also. And it was, as a refresher course, it was very detailed. So I found the topic very interesting actually.
Nasir, I have kept it very light. I've thrown some things at you so at least you know that somebody in the family is having an ApoA or ApoB. Why are they having it done? Okay?
Yes.
Or is it about this triad index? What is this? This is in children. It is something that it's the Indians who have discovered it. And it's interesting.
Yes, ma'am, we are not aware of this triad.
Mm-hmm.
Yeah.
So so it is good information for us also. We're not aware of these pentatriad.
Yeah.
Yeah.
In fact, we'll have to.
Unfortunately, we are not aware. Correct, you are.
Yes, that is the reason why when I brought this, I said I brought it for you all so that you know that something like this exists. And especially if you're working in a cardiac lab or cardiac hospital, these will come to you routinely. They'll come to you routinely. It's just not cholesterol and triglyceride. You'll get ApoA, ApoB, blah blah blah.
Ma'am, it's advisable then for the younger generation, like we have in our family, that they should undergo these tests like regularly. Now,
Well, if
since
it's in the family, then why not? Why not? Once in a while. Once in a while, yes.
like
Especially
how-
diabetes. If diabetes is there, you must monitor your fats. You must monitor your fats. There's no two ways about it. Anything else?
FOA, FOB is coming.
Huh?
Lipoprotein is also coming. FOA, FOB is coming. Lipoprotein A, lipoprotein B. But that was also one new concept, mutant of lipoprotein. That was also a new
That's interesting, isn't
the
it? Milano.
yeah, the
Milano,
concept.
yes, it's
That also we
interesting.
don't know.
That in
Yeah,
spite of
we
a
don't
in spite of a low HDL, there's no risk. There's no lining. There's no deposition in the arteries. That's
Ma'am,
wonderful.
are they synthetically manufacturing this? Like they give this treatment to lower the plaques or something like that? I think something.
There are infusions, yes. There are infusions, yes,
you
yes.
Discussed that, yeah.
In fact, I think it's abhi to nahi hai. There was one gentleman some years back who used to take these patients with high cholesterol, lipids, and all that, and he used to give them drips, and he has saved them from open heart surgeries. He saved them from open heart surgeries. The Muslim
This
gentleman.
topic is really interesting because now I'm thinking what happens with Monjaro. How does Monjaro is reducing the cholesterol over there? Where people take Monjaro to reduce the cholesterol and all these things. Reduce weight. So now I'm thinking, how does Monjaro work?
Is it a statin-based compound? That's what gets your cholesterol down. Yeah. It's a statin. Nazir, why don't you look it up how statins work? The different types of statins over here.
Also,
And
ma'am,
yeah, tell me.
there was something known as chelation therapy which people were doing.
Yes.
Some of them.
Yes. This is what I was talking about. You're right. You used the right word.
Yes.
And he used to give them drips and no open heart. They were fine.
Yeah, so you just go for a day and they give you a drip of this
Correct.
chemical, and then they say that it really reduces all your plaque and all. So is that true? It
Yes.
would happen.
That is what people said. In fact, somebody in Pune also does this.
Even in Bombay, we have somebody in
I told you that there's a Muslim gentleman who does this. Forgotten
yes,
his name.
Andheri or
Pune में भी है. The young
Okay.
doctor is there who's doing this, and no problem.
yes.
So, ma'am, if you have blockage,
Just
like,
tell your doctors.
Yeah. Continue.
no, it's okay.
If you have block,
Just tell your doctors to counsel you before.
pardon.
Or anybody should be counseled. Anybody who is going for these therapies, they should be counseled before with this.
Oh yes, you have to sign a waiver. You have to sign a waiver that they will not be responsible if something goes wrong.
Yeah, because I took one shot of Monjaro and I went mad because your brain will say something else, your body will say something else, and finally I had to leave it because I had to go join work. So you will feel hungry.
Why? But
but
why
you'll
you
not
went
feel
mad?
hungry.
Why you went mad? See, I'm on a statin. I'm on a very low dose statin. I take what's known as Rosley 5.
Oh.
That's
So this is
see, I have no trouble with my triglyceride. My cholesterol was a little high. समझे ना? At 220, 230 for years. Then eventually my diabetic specialist said, no, Rose, you have to get onto this and bring it down. I was very constant at 220, 230. Yeah,
I was feeling hungry, and at the same time you cannot eat also. You feel that you will be hungry, but you cannot eat. Your hunger will die.
is
So
it?
it is a mix, yeah.
I
The
don't
same
know.
thing happens to people who go for bariatric. Two people in my family have done bariatric, and one of them was not counseled properly. So he was under complete depression. because the first time you do bariatric, for months and months you cannot drink also anything. Only 1 or 2 spoons, then little half a little bit of 10 ml, 20 ml, 30 ml water like that. And then counseling is very important in these therapies. They do work, but I feel people should know about this concept also. Doctors, they should counsel properly.
No, I think you will sign a waiver, no, that you will not hold the doctor responsible. ये chelation therapy में ऐसा ही है.
Yeah, so in chelation, our GPs or anyone, they don't recommend it. They say this is all rubbish.
They will
But there
not.
are
I know
people
that.
who
I
have,
know.
the people who have done it have, it was an advantage for them. It has really worked for them. I know 1 or 2 people who have done it.
हाँ, ये जीपीज है ना, ये कोई रिस्क नहीं लेते. इधर तो इंजेक्शन लगने के लिए भी they are trembling. They don't even have 5% confidence of what injection they have to give. This is so horrible over here in this part of the world. You know.
No, I
They
think
don't even want to take even 1% risk for anything.
our doctors are pretty good over here. There's no two ways about it. The only thing the American concept of promoting a certain drug is there. You know, the drug walas have told them that if you prescribe my medicine to an X amount,
Yes, that's
I'll send you to Canada, I'll send you to London, I'll give you a
Yes.
car. All this is a very, very old thing. The same thing happened in pathology. You buy my machine, I give you a car. You buy my machine, I will send the whole family to Paris. It has happened. It has happened. Meena, you were saying something?
So, ma'am, I was just asking that if suppose somebody has a blockage,
Yeah.
like on the angiography,
Mm.
so you could use like this type of treatment to reduce the blockage, ma'am. This
See the very fact, Meena, go back. Angiography kiya, blockage mila. What doctors do, they are ready with the stents.
Yes,
So
yes,
they
ma'am,
will
are ready.
introduce the stent immediately depending on how many blockages you have.
No, no.
Otherwise, they will say go for a
A CT angio, a CT angio.
you know.
CT angio, ma'am. Not the routine angiography, the CT angio. Nowadays you have the CT machines, no, which are doing the angiography. So if it shows some 40% or 50%, so can one like go for this type of treatment?
I don't know. That depends on the individual. Meena, I cannot answer that question for you.
Okay.
Because regular angio में तो मिल जाता है तो फटक करके stent डाल देते
they
हैं.
put.
You know.
Yes, ma'am. Right.
Otherwise, I tell you, go for open heart. You got too many blocks. You know.
True.
Okay. Any other question? Parizad, anything you want to contribute?
Nothing, thank you.
Okay.
So very good, ma'am. Very informative.
Can we end because I have to go for a meeting?
Yes.
Yes,
It's a
ma'am.
school meeting.
Yes.
Yes,
And,
ma'am. Thank you
Next
so much.
Friday, what do we have, ma'am?
अभी क्या किया आपने? What is it?
Lipids and carbohydrates done.
I think it may be kidney function.
Okay.
Let's see. He'll tell you.
Okay. Thank you, ma'am. Really
Thank you.
enjoyed.
We'll tell you.
Thank you.
We'll give
Thank
it to
you,
you.
ma'am.
Yeah.
Thank you, ma'am.
Thank you, ma'am. Sorry for the delay.
I think it will be renal function, na? Renal function आएगा.
Renal function. Okay.
Yes.
Okay. Thank you, ma'am.
So we'll let you know. Righto. Thank you.
Okay, thank
God
you.
bless.
Thank you. Thank you.
Bye bye.
Thank you. Bye, everybody.
Markers.
We'll do liver function. We'll do this बाद में करेंगे मार्कर बाद में.
बंद कर दो ना.
Yeah.
चार्ज है ना. चार्ज खत्म नहीं हुआ.
No, you still got a fair bit. 10-15% है. देख निकला कितना है. Black, get the black cover over here.
निकालते हैं. फिर ये बंद कर देता हूँ.
बनकर फिर बाद में दर्द करती हूँ मैं. Cardiac and tumor markers will take last. इधर है
माउस काम नहीं करता,
माउस कैसा काम नहीं करता
थोड़ा
है
कम काम कर रहा है मैम।
because you hadn't pressed it all the way.